Mamosa, we are going to record this if that's okay with you.
Yes, it's fine.
Okay, so I'm just going to introduce you. So today's speaker is Mamosa. She obtained her BSc in microbiology and biochemistry from WITS, followed by an honors and master's degree in plant biotechnology at the University of Free State. She is currently a PhD student at the University of Cape Town and the University of Potsdam, Germany, where her research focuses on desiccation tolerance in Xerophyta resurrection plants. So outside her research, she is an avid runner. She currently is training for a 42.2-kilometer marathon, which is scheduled to take place in 20th October. So that's 2 weeks' time. She embraces the challenge of balancing rigorous research with her passion for running, believing that both pursuits foster resilience and determination. So I'm going to hand it over to you, Mamosa. Thanks very
Okay,
much.
okay, thank you so much. Can you hear me?
Yes.
All right, perfect. Okay, good morning, everyone, and thank you so much for this opportunity to share my research with you. Yeah, so the title of my talk this morning is Comparative Analysis of Desiccation Responses in Xerophyta Resurrection Plants at 2 stages of seedling development. As you all know, water is one of the essential elements required for plant growth and development. And in the absence of water, plants undergo a series of changes and effects that can ultimately lead to death. And one of the, okay, sorry, let me just get my laser point. Yes, perfect. Yes, so one of the effects that plants experience under dry conditions is the accumulation of reactive oxygen species, which is caused by continuous absorption of light during dry conditions. And this results in oxidative stress, which can severely damage cellular components such as proteins, lipids, and nucleic acids. Unlike animals which can move around and maybe run away when subjected to dry or stressful conditions, unfortunately, plants do not have that ability, but they have evolved adaptive mechanisms to cope with water limiting conditions. And one of the primary strategies that they employ involves morphological and structural changes, including closing their stomata in order to reduce water loss, which happens through transpiration. And they also enhance root growth so as to improve water absorption from soil. In addition, plants also accumulate osmoprotectants such as amino acids like proline and also some protective sugars as well, and this help in maintaining cell tegar. Although these mechanisms are effective to help plants with coping with water limiting conditions, the problem comes when the stress condition is prolonged because it results in significantly reducing the relative water content, and most plants cannot survive, unfortunately. However, there are some plants which have specialized adaptation strategies that enable them to survive under desiccation. And these are said to be desiccation tolerant. So basically, desiccation tolerance is the ability of certain organisms to survive severe dryness of up to 10% of relative water content and subsequently resume normal physiological processes upon rehydration. So desiccation tolerance trait is prevalent in orthodox seeds, which are produced by most angiosperms. And it is also acquired during the late maturation phase of embryo development. And notably, it is quite rare in vegetative tissues. But there is one exception, and that is resurrection plants. So they basically are this group of plants that have the remarkable quality to survive under extremely dry conditions and come back to life as soon as water becomes available. Hence the name resurrection, basically meaning that they can come back to life. And it only takes them a few days to resurrect, which is quite remarkable. And of course, the question that we ask is, how do these plants do this? How are they able to come back to life, even after being dried for several months to years? Well, studies have been conducted on resurrection plants to understand the mechanisms that they employ in order to acquire the vegetative desiccation tolerance ability. For example, what we know now is that the accumulation of proteins such as late embryogenesis abundant proteins, which help in protecting cellular integrity and function, is important for desiccation tolerance acquisition. And also, these plants also acquire or accumulate heat shock proteins, which act as molecular chaperones and protect proteins against degradation. They also accumulate early light inducible proteins, which are important in the protection of photosynthetic apparatus. And protect these from the damage caused by light. They also have effective antioxidant system in place, and this is important for protecting these plants during desiccation. Another important thing that I want to highlight is the two strategies that these plants employ. So they are grouped into two. One group is poikilochlorophyll, and the other group is homeochlorophyll. So I'll be talking a lot about this as we continue the presentation. So the poikilochlorophyll strategy is basically a strategy where these plants break down their chlorophyll and their thylakoid membranes during desiccation. And they do this because they want to basically protect themselves from the damage caused by reactive oxygen species accumulation. Because when plants dry down, they continue to absorb light, but because processes like photosynthesis are shut down, the continuous absorption of light can end up causing damage. So in order to overcome that, they just break down the chlorophyll, and this results in the change of the color of their leaves from green to yellow, which I will show you shortly. On the other hand, the homeochlorophyll group does not break down its chlorophyll during dehydration. So the chlorophyll and thylakoid membranes in the chloroplast remain intact. But what they do instead, these plants, is that they produce other pigments such as anthocyanins, which acts as sunscreens to protect the leaves against light. And they also fold their leaves so as to protect themselves against light. So this is the main difference between the two strategies employed by different resurrection plants. And now, as I've mentioned, I'll be referring to this a lot as I continue with my presentation. So the example of poikilochlorophyllous plants are Xerophyta schlecteri and Xerophyta humilis, which I'm using in my study. Yes, so Xerophyta schlecteri is native to parts of Limpopo and Mpumalanga provinces, here indicated in yellow, not yellow, blue dots. And then Xerophyta humilis has a broader distribution across the Southern African region. You find it in areas in Botswana, in Namibia, as well as in the northern parts of South Africa. So all these yellow dots are Xerophyta humilis. And then lastly, the example of homeochlorophyllous plants is Xerophyta elegans, which is predominantly found in the Drakensberg Mountains of KwaZulu-Natal and Mpumalanga provinces. Okay, so although these plants are very remarkable and interesting and unique, there are some challenges or the costs that comes with being desiccation tolerant. And one such challenge is prolonged period of growth. These plants take very long to grow. They can take several months to years to grow and start flowering. And this prolonged growth period may significantly limit the pace at which we conduct research. And then another disadvantage of working with adult plants is that the water content is quite variable across the plants. And another thing is that when you dry down the plants, the dehydration rates of plants can be complicated by the size of the pot as well as the amount of soil that the plants are grown in. So in order to overcome these challenges, my study employed a seedling model to investigate vegetative desiccation tolerance mechanisms employed by 3 Xerophyta species. And in my PhD project, I'm using omics approaches. I'm using transcriptomics, metabolomics, and lipidomics to further investigate the VDT mechanisms. But for the purpose of this presentation, I'm just going to focus on metabolomics and lipidomics results. So we performed GCMS and LCMS analysis to investigate desiccation-induced changes in primary metabolites, secondary metabolites, and lipid profiles in 3 Xerophyta species at 2 developmental stages. All right, so this is briefly the experimental setup. Okay, so first of all, we germinated and grown seedlings on half-strength MS media, and we incubated this at 23 degrees Celsius in a growth chamber. So we allowed this to grow until they reached a desired stage. So we had 2 stages of seedling development. Let me quickly show you. We've got the pre-leaf and the 2-leaf stages. So I'll be referring to them as early and late stage seedlings respectively. And this is how they look like under hydrated conditions at pre-leaf stages and 2-leaf stages. And then when they dry down, this is how they look like. And also these are the species that I'm working with. Okay, going back. Yes, so once the seedlings have reached the desirable stages, what we do is that we just transfer them from the MS media to a damp filter paper to conduct the drying down experiment. In total, we placed 20 2-leaf seedlings and 60 pre-leaf seedlings on each petri dish plate because these numbers were sufficient for RNA, or rather good RNA quality. So we ended up with 20 and 60 for 2-leaf and pre-leaf respectively. And then you'll also notice that in our box, so this is the setup that we use. In total, in the box, we have 8 petri dish plates with seedlings, some labeled RNA and some labeled LM, so that's lipids and metabolites. So we performed meshed data sets for RNA and lipids and metabolites. And we dried down this for 48 hours in a growth chamber, and then sampled and then conducted the extractions and sent the samples for GCMS and LCMS analysis. So now I will show you some of the results that we got. Okay, so first of all, we wanted to assess the clustering of primary metabolites, secondary metabolites, and lipids across 6 biological replicates. Yeah, I forgot to mention that we prepared 6 biological replicates. So we used PCA plots to do this. The separation of primary metabolites and lipids is not species specific. All these samples are mixed together. But now I want us to just focus on PC1 and PC1 in both. So the variation is quite low for both primary metabolites and secondary metabolites. However, there's a clear separation of primary metabolites between the early and late stage seedlings. Okay, apologies. I was supposed to explain the color codes so that you're not confused. So when you look on the far right, we have green. So that represents Xerophyta elegans. And then we have yellow-orange, which is Xerophyta humilis, and then blue, which represents Xerophyta schlechteri. And then the capital E represents the early stage seedlings, and then L is late stage seedlings. And then the shapes, so we have the round ones, which are dehydration, and then the triangles are hydration. I hope that is clear. Okay, so going back to this, although the samples are not clustered according to different species, we see that there is a clear separation of primary metabolites between the early, which are light in color, versus the late stage seedlings, which are darker. And then now looking at the lipids, based on the clustering in PC1. Okay, sorry, I just realized that it says PC1, PC1. This is supposed to be PC2. Apologies about that. So based on the clustering in PC1, it looks like lipid profiles in Xerophyta schlechteri and humilis, which I have circled here. It looks like they show similar patterns as they are grouped on one side, and they are predominantly occupying the negative side of the component, while Xerophyta elegans is mainly on the positive side of the component. And then now looking at the secondary metabolites, the clustering is clearly based on it's species specific, as we can see. There's elegance here. Oh no, I'm so sorry. So this is elegance. I see the labeling is wrong. The elegance, humilis, and schlechteri. Yes. So this basically suggests that each species has a distinct complement of secondary metabolites here. Okay, so now zooming into what actually happens within each species, we're going to start with primary metabolites. And the separation in PC1 across all these species is clearly based on developmental stages. So we have the early stage seedlings on the left, and we see this across all 3 species. And then the separation on PC2 is based on condition, with the hydrated ones in blue separating from the dehydrated ones in red. Okay, so now the question is, which metabolites are significant between hydrated and dehydrated seedlings? And to do this, we performed ANOVA analysis, and out of 78 primary metabolites that were identified, 23 of these from the early stage seedlings and 26 from the late stage seedlings were found to be significantly different in response to desiccation across all three species. And when we look at the early stage and the late stage seedlings, the general responses are the same. For example, there's an increase in the accumulation of sugars such as sucrose, which increases in dry tissues. And this is consistent across all species in both early stage and late stage. And we also see an increase in amino acids such as proline, which is also known for its role in desiccation response in plants. On the other hand, there are some sugars that are decreased in accumulation, and one such example is fructose, which decreases in response to desiccation, and basically the results are the same for both early and late stage seedlings. And the question was, what happens to glucose? Because glucose typically is expected to decrease during dehydration, as observed in other resurrection plants. But then in our results, there was no significant difference in hydrated and dehydrated seedlings. So it was not detected in the significant changes. But now I want us to focus on sucrose briefly, because it's one of the key sugar carbohydrates that plays a very important role in the acquisition of desiccation tolerance. And how it does this is that it has hydroxyl groups, which it uses as water replacement molecules. And this helps in the formation of glassy liquids in the cell. And it's very important for protecting cell components and ensuring that membrane is stable during dehydration. So it was quite exciting when we saw its accumulation in both early and late stage seedlings, because it's consistent with what we've seen in other resurrection plants. Okay, so that's basically some of the highlights from the primary metabolites, and now going to the secondary metabolites. So the general trends in the changes in primary metabolites are largely consistent across species, and it makes sense because primary metabolites are important for basic physiological function of plant cells, and they are highly conserved across different species. But secondary metabolites are often induced in response to environmental stresses, and they're important for protecting plants against different abiotic stresses, and their accumulation may vary between different species. Okay, so again, we wanted to look at the PCA plots to see the general patterns between the early and late stage seedlings, as well as the hydrated and dehydrated seedlings. And we see a clear separation between the two developmental stages and a segregation of hydrated and dehydrated samples. Okay, so we also wanted to know which secondary metabolites are significantly different between hydrated and dehydrated samples. And I've grouped these into clusters. So there's a cluster that I've labeled 1A and 1B, and then we have 2A and 2B. Cluster 1, A and B, basically just highlights metabolites that are constantly high in abundance in both hydrated and dehydrated tissues of elegans. So you can see here they are constantly elevated in fresh tissues and dry tissues. They are at high levels. But on the other hand, when you look at Schlecteri and Humilis, we see that these same metabolites respond in a desiccation responsive manner. This is visible in the early stage seedlings as well as in the late stage seedlings. And then the second cluster, which is cluster 2A and B, this one highlights secondary metabolites that are induced by desiccation across all species. So you can see that in fresh tissues or hydrated tissues, the levels are quite low. But then once the seedlings undergo drying conditions, then they start increasing. And the levels are, or the increase in these metabolites is consistent across all three species. Okay, and another one that I wanted to highlight is trehalose, which we see its increment as these tissues dry down in both the young seedlings and the older ones. So trehalose is known for its protective role in desiccation tolerance. And it's also, like I said, we see it in both early and late stage seedlings, and we also see it in adult plants as well of different resurrection plants. Okay, so we also asked the question of what is conserved across the species and what is different between these species. And we have found that a total of 10 metabolites, secondary metabolites, were found to be conserved. These are the upregulated ones only. So they were found to be conserved across all these species. But then we are particularly interested in those that are different because of the strategies that I've highlighted earlier, the poikilochlorophyll and the homeochlorophyll strategies. So now, just so you can see what I'm referring to, again, so the first strategy that I spoke about was that of plants that break down their chlorophyll. One example of that is Xerophyta schlecteri, which breaks down its chlorophyll and thylakoid membrane during hydration, and the leaves change from green to yellow, as we can see here. On the other hand, elegans retains its chlorophyll and thylakoid membrane, but does not lose its chlorophyll, right? And we can also see that the color of the leaves does not change to yellow, but it becomes dark green. And that's because of the accumulation of anthocyanin, as I've already mentioned, which act as sunscreens to protect the plants against dehydration. So among the metabolites that were only found in elegans were flavonoids, which have protective properties against ultraviolet radiation to protect chlorophyll. And then among those that were found between Schlecteri and Humilis and were not present at all in elegans were examples of dipeptides, and their accumulation mirrors the degradation of thylakoid membrane-associated proteins such as rubisco. So it makes sense why we find their accumulation in these two species and not this one. It's because these ones break down their thylakoid membranes while elegans retains it. Okay, so now moving to the last part of my presentation, we also looked at lipids. And Okay, briefly, lipids play a very important role in maintaining membrane stability and function. And they also form part of the thylakoid membranes in the chloroplast, which undergo several changes during desiccation. Okay, again, we did the same thing that we did in primary metabolites and secondary metabolites, where we looked at the general patterns in samples that have undergone dehydration and hydration. And again, we see a clear separation between the hydrated samples and the dehydrated samples, as well as the two developmental stages, the early and the late stage seedlings. And then regarding the changes in lipid profiles in response to desiccation, out of 132 lipids that were identified, 48 of these were significantly different in response to desiccation across all three species. And these were quite diverse, ranging from phospholipids, glycolipids, and galactolipids, just to mention a few. But in particular, I just want to highlight the last block here. And the reason why I'm highlighting this is because it's also going to help us understand the main differences between the two groups of plants that we have that break and do not break chlorophyll. So taking a closer look into this, we see that Xerophyta elegans has high levels of the two galacto. I don't know if you can see from your side, but we have two galactolipids here, MGDG and DGDG. So these are expressed at high levels in Xerophyta elegans at both hydrated and dehydrated conditions. On the other hand, these lipids decrease in levels in Schlecteri and Humilis. So under hydrated conditions, they are found in very high levels, but as soon as water levels go down, they also decrease in their levels. And this is consistent in both the species. And so that you have an idea of what's actually happening. So I also did transmission electron microscopy work where we actually wanted to see what happens inside the chloroplast with the thylakoid membranes in particular. So what we see here on the left is the structure of the chloroplast under hydrated conditions. We have runner stacks which are intact, everything is looking good, the plants are happy. And I also included this diagram so that it's even clearer. So we have grana stacks here, and then these are the thylakoid membranes. So thylakoid membranes make up grana stacks, and then these are found within the stroma in the chloroplast. So So these galactolipids, which I have mentioned here, right? These are mostly abundant in thylakoid membranes, okay? And what happens during dehydration is that in Xerophyta, Schlecteri, and Humilis, the thylakoid membranes are completely broken down, and this results in development of these vesicles. So all the grana stacks are gone. All the thylakoid membranes are completely dismantled, and all that we see are vesicles. And hence the change in the color to yellow, color of the leaves to yellow. On the other hand, in Xerophyta elegans, which does not dismantle its thylakoid membrane, it looks different because it doesn't have the vesicles. It still has the thylakoid membranes, even though they are not very clear on this image, but they are still present here. So these ultrastructural changes that we see correlate with the decreased levels of the two galactolipids, MGDG and DGDG, during desiccation in Schlecteri and Humulus. And this lipid breakdown of the two galactolipids highlights an effective adaptive strategy employed by poikilochlorophyllus plants in order to counteract dehydration stress imposed. Okay, so just to summarize, as mentioned at the beginning in my introduction, that the challenge with working with adult plants is that they take very long to grow and also variability in water content. So we have established a seedling model, which works very well, and it's an excellent model for studying desiccation tolerance mechanisms employed by resurrection plants. And the changes that we have observed in the accumulation of primary metabolites, secondary metabolites, and lipids, they play an important role in the acquisition of desiccation tolerance. And of course, there are many other things that accumulate that are expressed. We also saw in our RNA-seq results as well, but I only presented the metabolites work right now. And then lastly, relative to the effect of desiccation on the secondary metabolites and lipids, the three species under study exhibit strong species-specific and, to a lesser extent, developmental stage effect. So this is basically what I prepared for this morning. Thank you so much. All thanks to my supervisors for their guidance, as well as other people who made this work possible and funding. Thank you so much for your attention.
Thank you so much for the presentation. It was a very good presentation and a few hiccups here and there, but you can see you did a lot of work for your PhD, so well done.
Thank you.
So I'm not much of a plant person, so I'm going to ask anyone in the audience, are there any questions for the speaker?
I have one. It's
Go
very
for it, Malati.
very generic question. So the challenge that the plants take way too long to grow, how does this affect I guess the thinking with studying resurrection plants was if we understand their mechanisms, then that could be applied in crops and improving in food safety and security. How does that affect that ideology then of maybe applying these mechanisms if they take too long to get back up?
Okay, so unlike other plants like Arabidopsis, which takes a few days to germinate and grow, these ones do take long. But then and obviously, if you want to do a lot of studies, it means you'll have to wait for a very, very long time in order to get some of your questions answered. And that is inconvenient, right? Because I remember at the beginning of my PhD, we were actually thinking of germinating these plants from scratch and having them to grow until they flower. And then we realized later on that this was going to take forever to grow. So I think with the question that you're asking, I think if we select some genes and then incorporate them in crops which are already mature, I don't think that would be too much of a problem because they would have already attained the desired phase of growth that you want them to be. Unlike maybe if we start them from scratch, we don't know, maybe that might also affect their growth. I'm not sure about that. I haven't looked into that, but I'm just thinking right now that maybe if, for example, we incorporate some of the genes from at a seedling stage, maybe in maize or rice, for example, it's possible that it might affect the growth rate. However, if we introduce them when they are already mature, then maybe that will be different. Yeah, that's what I think.
No, I hear you. I hear you. I guess if you're applying one aspect of
Mm,
the resurrection mechanism, then you're not
mm.
necessarily taking over the plant's whole physiology. So it will still probably keep to its characteristics of
Yeah.
if it did. Okay. Thank you.
All right.
Wilson,
All
I
right.
see you have a question.
How do you know? Okay. All right. Well, thanks for the presentation. That was nice. At least we a better understanding of xerophytes now. Uh, I just want to ask something. Of course, you didn't discuss about transcriptomics. I see
Mm.
that you did some transcriptomics work, right?
Yeah, I did.
Okay. I mean, just out of curiosity, how did you ensure that the RNA was not degraded? Because I know RNAs are less stable than DNA.
Yeah.
So how did you ensure that they are still intact before your analysis?
Yeah, that's a good question. So obviously that took a lot of optimization of the protocol, especially because these plants are non-model plants. So we had to be, with the protocol that we're using, we had to be very, very careful, with like technical stuff. But also once we were able to get good quality RNA, we had to transport it to China. So we used these tubes. I think I forgot their names, but once you put your RNA in them, it's quite stable and it can travel for long distance without any issues. I just forgot the name of the tubes that we bought, and they were quite helpful in making sure that our RNA is stable and doesn't degrade.
Did they have a special design or something?
You mean the tubes?
Yeah.
Ooh. Okay, let me see if I can find the name quickly. But they were just normal tubes with something that looked like a filter inside to absorb the liquid. And then we had to dry them down on speedy vac so that, before we send them off for sequencing. And then when they got there, they suspended them and then, did a whole lot of processes first. But I'll try to find the name and maybe I'll type it in the chat when I find it.
It's okay.
So,
Thank you.
Donald, do you have a question? No. Okay, can you go back one more slide, Mamosa?
okay, wait. Which one?
Okay, no, that one.
This one.
The next one. I just want to leave it up there. Can you also put your email in the chat box? I just like to have this open, so if anyone wants to contact you or your supervisors
Oh, okay.
regarding the work that you do, so that any follow up from the video online on YouTube when we upload it, we always like to make it the presenter available.
Okay.
Hi, Donald, do you have a question? I see you unmuted.
Okay, yeah, so I've typed my email address in the chat.
Okay, so your email is ngcmam002@myuct.ac.za.
Yeah.
Okay. So if anyone is listening to this after this current presentation on the YouTube world, if they're interested in any of the work that presenter is doing or wants to get involved with this group, we encourage them to contact either presenter directly or someone in the group here. So you can Google them and see what they are doing and get involved.
So, Shane, you should maybe
Yes.
start recruiting her for your Northwest team there. They do a lot of marathon metabolomics so that she she
Oh,
doubles in what she already loves.
we do veteran runners. So this is the first time that she's running. So maybe after 3 marathons, we'll get in contact with her again.
But I've been doing a lot of halves, so
There we go. Okay, so yeah, something to do for postdoc to keep in mind.
Yeah, if you want
Marathons.
to do research in marathons, yeah, contact us. Prof. du Toilet, he's a specific person.
Mm.
Mm.
Okay.
And you've already been all over the country, you might as well add Northwest there.
Why not?
Right.
Yeah, good luck on your marathon on the 20th.
Thank you so much.
I hope it goes well.
I hope so too.
And good luck with the rest of your PhD.
Thank you so much. And thank you for the invite to share my work with you guys.
No, thank you. That was a great presentation.
Thanks.
Okay, I think we can end it there, Maloti.
Okay.